All right, guys, if you understand this slide right here, you sort of understand meiosis, at least in general. Let's look real close. This is the big picture. In a second, we're going to get to all the nitty-gritty details, but let's start with the big picture. Here's a cell that's diploid. 2n equals 4. Remember, that 2n means diploid, right? If this was your cell, it would say 2n equals what? 46. But this is a diploid cell with 4 chromosomes. So that means that it really has how many different kinds of chromosomes? 2, right? Because diploid means it's got 2 of each kind, right? So its haploid number is n equals 2. But this is a diploid cell. What hasn't happened to these chromosomes yet? They hadn't been replicated. Because if they were replicated, they would look like what? They'd look like X's. So these haven't been copied yet. Between here and here, you can see they have been replicated, right? So between here and here, replication happened. They became what kind of chromosomes? Bivalent. Bivalent chromosomes. X-shaped. So up here, again, if this was your cell, first of all, what kind of cell is that? What are the cells that can do meiosis called?
Germ cells.
Germ cells. Germ cells are only found where in your body?
In your ovaries and your testes.
In your ovaries or your testes, hopefully, right? Not in your ovaries or your testes, right? One of the two. So let's do this for a human. If this was a human germ cell, it would have 46 chromosomes in it right here, right? And right now these chromosomes are single-stranded. They'll be like that. What do we call the shape of our chromosomes? Eukaryotic chromosomes are linear, okay? They're not really straight lines like that picture shows you, but they're definitely not what? Definitely not circles. They're not loops. Here, though, they're single-stranded. We got 46 of these guys. Between here and here, replication happened. So now in our germ cells, we have 46 bivalent, X-shaped chromosomes. Meiosis happens in 2 parts. Meiosis 1, meiosis 2. In meiosis 1, the partner chromosomes get split apart from each other. What's the word for the chromosomes that are partners with each other? It's the same kind of chromosomes. What are they called? Heard somebody say it, homologous. Or sometimes they're called homologs. Now realize, when we're saying that they're the same kind of chromosome, that doesn't mean they're exactly alike. It means they've got the same what on them? Same traits. They control the same traits. They have the same genes in the same order, but they may have different what for those genes? Different alleles, different versions. But what we're saying, like in these little chromosomes right up here, they use colors here to show you that one of these came from where? One came from mom, one came from dad. If the first gene on this chromosome was for eye color, the first gene on its homologous chromosome is also for eye color. But they may have different alleles. One may say brown, one may say blue. They're not exactly the same. In meiosis 1, though, it's the homologous chromosomes that separate from each other. So one of these goes one cell, one goes into the other cell. Homologous pairs separate. So after meiosis 1, we get 2 cells. In a human, how many chromosomes would be in here? 23. And there'd still be X-shaped, right? 23 X-shaped over here. We're trying to get 23 chromosomes in our sex cells, right? Why don't we stop there? What's still wrong with those cells, with those chromosomes? They got bivalent chromosomes, right? Is that what we want for our cells to work the right way? No. To work, we need cell division, whether we're talking about mitosis or meiosis, whichever one we're talking about. The cells at the end should have single-stranded chromosomes. One molecule of DNA, not 2. We don't want bivalent. We want single-stranded chromosomes. So meiosis 2's job, the second part of meiosis, is to take these X-shaped chromosomes, the bivalent ones, and separate them. Meiosis 2 is pretty much just like mitosis. Not really any different. Meiosis 1 takes diploid, halves the number of chromosomes. Meiosis 2 takes bivalent chromosomes and splits them in half. It's just like mitosis. So we start with one diploid germ cell. And we end up with 4 cells down here at the bottom that are haploid, and we're going to call those cells eventually gametes or sex cells. If you understand that, you got a big picture of the way this works. And we want to go a little bit deeper with this, though. Let's skip the video for right now. These terms you should already know. We know diploid means 2N, that you have 2 copies of each kind of chromosome. Haploid means N, and that means you have 1 copy of each kind of chromosome. The only haploid cells in your body, in the human body, should be what? Your sex cells, your gametes. Now, is that true in every other living thing? No. There are some things that are usually haploid. Like what that we just talked back a few weeks ago? What kind of organisms did we talk about a few weeks ago that were almost always haploid? Well, bacteria are true because they only got 1 chromosome to begin with, right? Fungus. Fungus are always haploid except during reproduction when they become diploid. Sort of opposite of the way we work to some extent. We talked about somatic cells, and we said those are what? Body cells, not gametes. Diploid body cells in humans. But with meiosis's main job, its main role, is to create haploid gametes from diploid germ cells. And that's important, because if we didn't do meiosis to make sperm and eggs, what would happen to our number of chromosomes every generation? It would go up. It would double every generation. What do we know just having 1 extra chromosome typically does to a zygote? Most of the time it kills it. If it doesn't kill it, it causes it to have some kind of severe physical handicap or mental handicap. So meiosis helps to keep our chromosome number the same from generation to generation. That's its main job. Its other job is to create variation. I asked you guys this question the other day. Could your parents theoretically have your identical twin 9 months from now? Theoretically, they could. But it turns out it's more than a 1 in a billion chance. It's almost impossible. Because meiosis is designed, or let's put it is either designed or it evolved to create bunches of different gametes in each parent.
So what happens when you have a sister that's like 18 and then you have like a little sister that's 12 and they look exactly alike?
They can look a lot alike, but if you did a genetic screening on them, they wouldn't be identical. They got a lot of the same genes, but not every gene's the same. What I'm saying is that it turns out that, and we'll talk about more of the numbers in a minute, but each one of you can make somewhere in the neighborhood of nearly 1 trillion different sperm or 1 trillion different eggs, genetic combinations of them.
Women want all
They are, but I'm saying the mathematical combinations that they could make, there's something like 1 trillion. So the chances of your parents having the same exact sperm and the same exact egg and then getting together a second time would be like over 1 trillion times 1 trillion. It could happen, but in the real world it ain't going to happen, right? It's not going to happen. It could, but it's just not going to. So meiosis's job is to make sure that kind of thing doesn't happen, because variation is the foundation for what? And evolution. It's how evolution works. We need variation to evolve. So meiosis is one of the most important things for giving us that variation. Now, here's our life cycle. And this we'll touch on the twin thing here in a second. We start out as haploid sperm, haploid egg. In people, there would be 23 chromosomes in here and 23 in here. 23 and 23, hopefully, give us a diploid zygote with 46. 2 of each. That zygote will undergo mitosis very quickly. What's that initial ball of cells called that forms after the zygote being made? The blastula. And that's going to start to differentiate and move into germ cell layers. And what's it called once it's organized itself into 3 layers of cells? The blastula becomes the gastrula. And that gastrula has an opening in it called the blastopore. And that blastopore in you becomes the anus. And also in like echinoderms, it becomes the anus, right? We're called what? Since the anus forms first. Deuterostomes. Most of the other animals are protostomes. Their mouth forms from that blastopore, that hole. But regardless, mitosis makes all the different cells, which means every cell in your body genetically is identical to that first cell, right? Every cell in your body is genetically identical to that zygote. If the zygote was messed up, every somatic cell in your body is messed up. That cell is pretty important. So we continue to grow by mitosis. We grow into either a diploid adult female or a diploid adult male. Do meiosis, go from diploid to haploid, and start all over again. In both periods yesterday, somebody asked me about twins, like identical twins. In identical twins, one sperm fertilizes one egg. You get one zygote. For reasons we don't exactly understand, what then happens to that zygote? It splits by mitosis into two separate zygotes that are genetically identical. This grows into one blastula. This grows into another blastula. You get two identical babies. Now, if we're talking about fraternal twins, it's two different sperm, two different eggs, grow into two different blastula. Fraternal twins are no different than brothers and sisters born years apart. It's just happened that the woman happened to release more than one egg a month. Typically, a woman releases one egg per month, right? One egg per menstrual cycle. Occasionally, the body will mess up and it'll release more than one.
So when it releases more than one, doesn't that mean that she starts to, like, her eggs start to decrease?
Maybe slightly faster, but every month, several eggs start to develop. It's just most of them die before they're ever released. Yeah.
So, like, is that how this one of the twins had a baby that was like from one day and then had a twin?
Somebody always asks that question. Yes. I'll start with this. Dogs, for example. You ever notice a litter of puppies and how different they sometimes look from each other?
Yeah.
It's very common for a female dog to have puppies in the same litter with different daddies, 2 or 3 different daddies. Because she releases several eggs, and you guys know how it works in dogs. Every male dog in the neighborhood finds that female dog during heat time, all right? That's the way it works, okay? That can happen in human people as well. Okay?
But that's like
It's really rare, okay? Really rare, but there are documented cases where a woman had sex with 2 or 3 guys within, a day, and she got pregnant by 2 different people at the same time. Okay? It happens. Okay? I mean, there's a very small window.
It said that it can only happen with 48 hours or less.
Right. There's a very small window during the month where a woman can get pregnant. So it would have to be in a very short time span for that to happen. But it could. It's possible. We know it's happened. It's not as rare as you might think. Anyway, let's move on here. We need to talk about the different stages of meiosis. And this is something you got to understand. It's going to be on the quiz on Thursday. So bear with me. Meiosis, remember, happens in 2 different sets. Meiosis 1, meiosis 2. The nice thing about it, in both meiosis 1 and meiosis 2, the stages are pretty much the same stages we had in mitosis. Prophase, metaphase, anaphase, telophase. But they happen twice. So we use Roman numerals. There's prophase number 1, and then there's metaphase number 1. Then anaphase number 1, then telophase number 1. And then we switch over and we do the number 2s. Okay? But the first part, we start off with a cell after S phase of interphase that is diploid, and the chromosomes, since they've already been copied, look like what? Bivalent, egg-shaped. Now in this presentation, I'm using this word mitotic. That's kind of the older word for bivalent. We're going to use the word bivalent. Egg-shaped chromosomes. 2 chromatids. So after S phase of interphase, in a human, your germ cells would have 46 X-shaped chromosomes. That's what we mean right here. Diploid with 46 X-shaped chromosomes. Okay, thank you.
Can I go take that down to Miss Johnson?
All right, now, meiosis 1 starts. Meiosis 1 is called a reductive division, and that's a term you need to know, reductive division. Because meiosis 1 is going to take that germ cell with 46 bivalent chromosomes, and it's going to split it into 2 cells with what? 23. It's going to halve the number. It's dividing the number in half. It's reducing the number in half. So meiosis 1 is reductive division. At the end of meiosis 1, you're going to have 2 cells that are now haploid, but they're still going to have bivalent chromosomes, X-shaped. The first stage of meiosis is prophase 1. And it turns out prophase 1 of meiosis is a lot like prophase of mitosis. Let's talk about the things that happen in prophase of mitosis. What happens with the nuclear envelope? It dissolves because it got phosphorylated, right? What happens with the spindle fibers? They start to form, and what's the structure called that forms them? Centrosomes. What happens with the nucleolus? It dissolves. What happens with the chromatin? Winds up and makes chromosomes, right? Chromatin winds up and makes the chromosomes. That same stuff happens here. Same stuff that happened in mitosis. But there's an extra step. The extra step that happens in meiosis is really important that you know. It's called synapsis. Now we've used that word before, a similar word. Where have we talked about the word synapse before?
In the brain cells.
It's a connection between 2 nerve cells, right? Synapse means to join. That's what the word means, to join. In meiosis, the homologous chromosomes join together. So we would have how many pairs of homologous chromosomes join together here? How many pairs of homologous chromosomes do you have in your cells? 23 pairs, right? 23 pairs gives you 46 total, right? The homologous chromosomes find their partner and they wrap around each other. That's called synapsis. They join. When they join together, and I'll show you more detail on the next slide, but sometimes they swap pieces. A piece will break off and they'll switch them. So let's think for a second. If this was 2 homologous chromosomes right here, one from your mother and one from your father, like so, they're homologous. They've got the same genes on them, but probably different alleles. Right now they're joined together, they're synapsed. These points where they join together at have a name, and it's a name that AP people like to ask us about. These points where the 2 homologous chromosomes are joined together are sometimes called chiasmata. What I'm saying though, once they're joined, when they get ready to separate, they sometimes break. Where 2 homologous chromosomes join together. And what we end up with is something like this. Where that chromosome looks like that, and its partner now looks like this. Now let's think about what this means. Let's say the blue chromosome originally this was happening in you, and the blue chromosome you originally got from your mom, and the red one you originally got from your dad. After these things have synapsed and switched pieces, now what's in you? What's that chromosome? It's part what you got from your mom, but also part what? What you got from your dad. It's created a new chromosome. It's created a new chromosome that you don't have in your somatic cells. This is happening in your germ cells only, right? This chromosome you can pass on to your children, but you don't actually have it yourself. What is that doing for genetic variation? It's increasing it because it's taking DNA from your mom and DNA from your dad and it's mixing them together. It's shuffling the genetic card deck. It's recombining your genes. It's a point where the homologous chromosomes join together. So it'd be like right here and right here. So that process of homologous chromosomes switching segments is called crossing over. When they join, it's called synapsis. When they switch segments, it's called crossing over. That's one of the ways that meiosis creates this genetic variation that we have. So homologous chromosomes join at places called chiasmata. Segments break off of each one. They switch places. It's called crossing over. Every time that happens, that creates a new chromosome. A new chromosome that you don't have in your normal somatic cells, but you can pass it on to your babies. That's really important. Let's look at the next slide and we might see this a little bit more. Here we see, this is prophase 1. Here we see those homologous chromosomes all wrapped together. That's synapsis, where they join. And remember, if this was you, there would be 23 pairs of these guys all wrapped together like that. We'll come back and watch that video in a second. Here are 2 chromosomes that are joined together. These points would be our chiasmata, and they're about to cross over. They're about to switch pieces, switch gene segments. Here you can see where, again, they're synapsed right in here. And you can see their chiasmata, where they're joined together. And then what ends up happening after crossing over is that we've got new pieces of chromosomes. Pieces of chromosomes that you yourself don't actually have, but that you can pass on to your babies. Chromosomes that are part of a chromosome you got from your mother, and part of a chromosome you got from your father.
So is that where the variation comes in?
That's where a big part of the variation comes from. It's mixing up your genes. It's making new versions of your chromosomes. I'm going to stop the video there for just a second because I want to show you a little video clip. Okay, so after prophase 1, we get metaphase 1. Another important point. Metaphase, what always happens during metaphase? They line up. Now, in meiosis, it's important that you understand how they line up. In mitosis, they line up single file, right? In meiosis, they don't line up single file. They line up in 2 lines. Who lines up across from whom? What's the word for pairs? The homologous chromosomes line up across from each other. So you're going to have 2 lines of chromosomes. One over here, one over here. They're going to be lined up in the middle. That's the first key thing to know, that in metaphase 1, you get 2 lines of chromosomes. Homologous chromosome across from its partner. So in us, we would have how many pairs lined up that way? 23. Now the other key thing, and this is important, really important, when they line up randomly. What I mean by that, remember, one of these came from your mom and one of these came from your dad. One's maternal and one's paternal, right? Maternal from mom, paternal from your father. When they line up, it's random. Moms can line up over here or over here.
But they still have the same function.
Same basic function, but different alleles, right? There's 23 pairs. How many different ways can we line up 23 sets of 2? How many different ways can you match up 23 sets of 2? Turns out a whole lot. Turns out 2 to the 23rd power. How many times is that? Anybody have a guess as to what 2 to the 23rd power is? No calculator, just take a guess. What do you think? 2 to the 23rd power is 8 million and something. Okay? 8 million in something different ways. And that's important because once we line them up, again, moms could be over here or over here. The next time around, moms might be here, dads might be maybe down here. And it's random every time. So every time you get a different combination, ladies, let's pay attention, please. And then what's eventually going to happen to this set up here and this set down here? What's going to happen to these two sets of chromosomes in the next stage, anaphase? What always happens in anaphase? Things what? Split, separate. So this set's going to go one way, and this set's going to go the other. So what I'm telling you, that process I just described of randomly lining up across from each other, that's another way to get variation. Because every time, how do they line up? Differently. And they end up in different cells. That process that I just described to you is called independent assortment. And that term confuses people. But it's something they're going to ask you about on an AP exam. You need to know how to explain it. First of all, know it happens in metaphase 1. And know it's when homologous chromosomes randomly line up in the middle of the cell across from their partner. And that sometimes the chromosome you got from mom lines up on one side, sometimes it lines up on the other side. It's random. So that every time those chromosomes line up, you get a different combination. Because how many different combinations did I say you could get? 8 million and something. So it's a crazy amount of variation that this can make. Here's a really technical definition of it. When maternal and paternal chromosomes are lined up on the opposite sides of the metaphase plate, middle of the cell, in a random fashion. And these are going to then split. It's a way to create variation. Know what independent assortment is. That's the main thing that happens in metaphase 1. Chromosomes line up in the middle. They line up across from their partner. They line up randomly. So that every time they line up, essentially you get a different combination. Because there's 8 million different ways for it to happen. Metaphase 1. Now after metaphase 1, after metaphase always comes what? Prophase, metaphase, anaphase 1. In anaphase, things split. What's another word for split or separate? What word do we use when we split the races up in the South? Segregate. Segregation. We're going to use that word again here. Segregation means to separate. In anaphase 1, you need to know we get segregation. What are we separating in anaphase 1? What are we separating from each other? What chromosomes? What did we just say we're lining up, getting ready to separate? The homologous chromosomes. We're going to separate each homologous chromosome from its partner. That's what segregation is. Segregation happens in anaphase 1, and it's when homologous chromosomes are separated from their partner. We're not separating the bivalent chromosomes right now. We're not breaking apart the X's. We're separating one X from his partner X. That's what happens in meiosis 1. This X gets separated from his partner down here. This X gets separated from his partner. In telophase 1, remember tele means far away. I'll get you in just a second. In telophase 1, those two sets of chromosomes get to opposite sides of the cell. Telophase 1 is pretty much just like telophase in mitosis. What happens around each set of chromosomes? What do you get around each set of chromosomes? Once they get to opposite sides? You get a new nuclear membrane. What happens to the spindle? It goes away. What happens in the middle of the cell? It starts doing cytokinesis and it splits in two. In humans, after meiosis 1, we would have two cells. Each with how many chromosomes? 23. We split the partners from each other. So we're down to 23. What shape are those chromosomes still after meiosis 1 is over? What shape are the chromosomes?
Bivalent.
Bivalent, X-shaped. And that's where we stop with meiosis 1. Janae, you had a question before we go.
The next Victoria is still, you said place apart from the partner, are they still X's?
Yeah, this set of X's moves one way, and this set of X's moves the other. Yeah, that happened in prophase 1. That happened a long time ago. But these guys, we're not breaking up X's in meiosis 1. we're separating one X from the other X. We're not actually separating the X's into single strands.
So is that after they do one split, they're
The split. Right. And we're going to get to more details in the meiosis 2 about how the rest of that happens. But after telophase 1, we have a very short period called interphase 2, or sometimes it's called interkinesis. So right after telophase, we get interphase 2 or interkinesis, but it's very short. What do we not want to happen here that usually happens during interphase? What's one of the most important things that usually happens during interphase? DNA what? Replication. If that happened here, we just undid all the work we just did in the first part of meiosis, right? So, okay, normally during interphase, the DNA gets copied. We just split a cell with 46 chromosomes down to 2 cells with 23. So, if we copied it again, we'd be back to 46, right? We don't want to do that, okay? Because what are we trying to get down to?
23.
Meiosis is trying to get down to 23 chromosomes with what shape? Single-stranded, not bivalent, right? We're halfway there at this point. We're down to 23, but what shape are the 23 still in right now? They're still bivalent. Part 2, which is going to happen right after interphase 2, is going to take those bivalent chromosomes and split them into single strands. That's what part 2 of meiosis is all about. So far, what we did is we started with one cell in humans with 46 X-shaped chromosomes. Meiosis 1 happened. It split that in half. It mixed them up, did all the crossing over and independent assortment, but we ended up with 2 cells, each with 23 X-shaped chromosomes, bivalent. Meiosis 2, which we're about to explain, is going to take both of these and split them again. Meiosis 1 separated what from what? What kind of chromosomes? Homologous. Meiosis 1 separated each homologous chromosome from its partner, but they're still X-shaped. Meiosis 2 is going to take these X-shaped chromosomes and break them into single-stranded chromosomes. And eventually, after meiosis 2, we're going to end up with 4 cells, each with 23 chromosomes. Of what shape chromosomes? Single strand, which is what we need for them to work. So let's go to part 2 of meiosis. Let's go through these pictures first. Here's metaphase 1. In metaphase 1, remember, homologous chromosomes line up in 2 lines across from their partners, and they line up randomly. What do we call the random alignment of chromosomes during metaphase 1? Independent assortment. Independently assort. They independently line up. All right, in anaphase 1, we can see the X's aren't getting split. This X got separated from his partner. This X got separated from his partner. And we call that separation segregation. That happens during anaphase 1. In telophase 1, they made it to opposite sides. All the normal telophase stuff happens. New nuclear envelope, chromosomes unwind, spindle goes away, pinches in the middle. We've got 2 cells. If this was happening in a person, there would be 23 of these X-shaped chromosomes in here and 23 in here. And that takes us to meiosis part 2. Turns out meiosis part 2 is just like mitosis. There's no extra stuff like crossing over, independent assortment. It's really straightforward. Meiosis 2 is simple. Realize though, at the end of meiosis 1, you now have 2 cells. 2 cells with how many chromosomes? 23. What shape chromosomes? Bivalent. It's the job of meiosis 2 to take those haploid cells and to take the chromosomes that are bivalent and split them into single-stranded chromosomes. That's all meiosis 2 is about. Breaking up the X's, splitting them into single strands. So in prophase 2, the normal prophase stuff happens. Nuclear envelope dissolves. Nucleolus dissolves. What forms? Spindle fibers. What else forms? Chromosomes. Chromatin winds up, makes some chromosomes. It's the normal stuff. In metaphase, same stuff that normally happens. What always happens in metaphase? Yeah, but the main thing, they line up. Chromosomes line up where? In the middle, metaphase plate. In metaphase 2, how many lines of chromosomes do you think we have per cell? One line. Because there are no what left? What did we separate away from each other during part 1? We separated up the partners. They're different cells now. There are no partners. In us, in metaphase 2, we would have one line of how many chromosomes? 23. And what shape are those chromosomes? Bivalent. Okay, they're linear, but they're bivalent. So we're going to have one line of 23 X-shaped chromosomes in metaphase 2. In anaphase 2, what always happens in anaphase? Something gets separated from something else. In anaphase 2, what's getting separated from what? The bivalent chromosomes are going to be broken into their two what? A bivalent chromosome is made of two chromatids. Okay, they're going to get split. The sister chromatids get split. And they start moving toward opposite sides of the cell. In telophase 2, the normal stuff happens. They make it to opposite sides. What happens to the chromosomes? What always happens to the chromosomes during telophase? They unwind and go back to being chromatin. What happens to nuclear envelopes? They reform around each set of chromosomes. What happens to spindles? They disappear. What happens to nucleoli? They come back in each new nucleus, right? And then we get cytokinesis. Remember, this was happening into two cells. So if two cells split, we end up with 4 cells. And in humans, these 4 cells would now be haploid, and they would have how many chromosomes? 23. And what shape would these chromosomes be? Single-stranded linear chromosomes, not bivalent. Now I want to walk you through some pictures of this. Here's the cell going into prophase 2. Now remember, if this was you, there would be 23 of these X's in each of these 2 cells. Because we're N equals 23, right? In metaphase 2, those chromosomes line up. So again, in you, in each cell, there would be one line of 23 X's. One line of 23 X's. In anaphase 2, the bivalent chromosomes get split. One set going this way, one set of chromatids this way, one set this way. One set going this way, one set going that way. It's happening in 2 cells at the same time. Yeah.
Okay, so you know how they have crossing over, how they cross over with the different chromosomes?
Mm-hmm.
Well, wouldn't it be 2 of them in the same part?
Wait a minute.
Like that one right there is just like the top one.
Let me go back. You're saying right up here.
Yeah, like it already has one that's crossed over, but why wouldn't it have another one now?
It should. I don't know why they don't have it. They should have a.
It's on the other cell.
Oh, yeah, okay, yeah, it's over here. Because remember, it's split in half.
So it wouldn't be another one at the top?
No, not necessarily. Crossing over is random. It doesn't happen to every piece of every chromosome every time. So there's the other crossover piece right there. Telophase 2, those sets of chromosomes made it to each side. And remember, in you, there would be 23 here, 23 here, 23 here, 23 here. And notice they're single-stranded. Then we get cytokinesis. We end up with 4 cells that are eventually going to be what? Gametes, sex cells, haploid sex cells. Now, here's something we haven't talked about. Meiosis is one part, the first part, of what's called gametogenesis. What's gametogenesis mean?
Beginning of being.
Not beginning. What's genesis mean?
First.
Creation. Genesis means the creation. So it's the creation of gametes. In us guys, gametogenesis in guys is called spermatogenesis. Makes sense? Creation of sperm, right? Now, I'm gonna go away from this slide for a minute and show you a picture. Spermatogenesis, creation of sperm. Us guys sort of follow the rules when it talks about spermatogenesis. We start with these cells called primary spermatocytes. What have we been calling those cells up to this point? Those are what? What? What are the cells we start meiosis with? Germ cells. Primary spermatocytes are guy germ cells. They're diploid. Remember, key point. You guys that are reading books, so I put those up. It's not English class. We're starting with how many chromosomes inside these germ cells? If we're in a human, 46. And these chromosomes are going to be what shape, assuming replication's already happened? 5 What you got, Dan?
But isn't it in these notes?
They're in the
I can't
outline.
find anything that you've been saying.
Are you looking at the mitosis or the meiosis?
I'm looking at meiosis,
Yeah, it's all in there. You're looking at the,
the outline.
it's in there. Yeah. You may not be looking at the right one. I don't know. We'll have to look at it.
Chapter 13.
I don't know. 46 X-shaped. They go through meiosis 1. Remember, meiosis 1 is about separating what? What gets separated in meiosis 1? Homologous chromosomes get separated from their partners. So we go from 46 X's down to what in these two cells? 23 X's. 23 bivalent chromosomes. All right. Notice these cells are now haploid because they've only got one of each kind, 23. They're called secondary spermatocytes. These guys then both undergo meiosis 2. And remember, meiosis 2 is about separating what? What's meiosis 2 separate? Each bivalent into single chromatids, right? So these split. What do we end up with in each one of these? 23, but they're not X's anymore, right? They're single strands. We got 23 of those. We got 23 of those, 23 of these, and 23 of these. Those then, that's the end of meiosis. Meiosis ends with these cells called spermatids. But they have to grow up. There's a part of the testy called the epididymis. I may misspell this here. It's an I think. Epididymis. That's a part of the testy where these guys sort of grow up. They mature and they become sperm. But that's the way it works in guys. In guys, one diploid germ cell leads to the creation of 4 working haploid sperm.
Why are they different, like, sizes?
All right, well that's the key. They're showing these different shapes because are those 4 sperm the same?
No,
They're genetically different. That's why they're showing those different shapes.
All of them?
They're all genetically different because of independent assortment and what? Crossing over. They're all different. Remember, a guy can make like way over 8 million different kinds of sperm. The 8 million just takes into account independent assortment. That doesn't even touch on what? Crossing over. So we're talking probably trillions of different sperm actually that each guy can make, genetically speaking. Now you ladies tend to have a way of complicating things, right? In women, it's called oogenesis. Anytime you see 2 O's together in biology, that means eggs. Okay? So it's the creation of eggs.
like with our oviparous,
It has something to do with ovaries and eggs. But if it starts with an O in biology, OO or OB, there's a good chance it has something to do with eggs. Oviparous, ovoviviparous, exactly. Oogenesis, though, is creation of eggs. Now here's the thing. In men, one diploid germ cell becomes 4 working eggs. No, that's wrong. One diploid germ cell, that'll be a weird man, right? One diploid germ cell becomes 4 working sperm in a man. In a woman, it's more complicated. In a woman, that one diploid germ cell does split into 4 cells.
that doesn't make 4 eggs.
What happens to 3 of those cells? They die. Those 3 that die are called what?
Polar bodies.
They're called polar bodies. What's their job? Why bother if they're just going to die? What are we trying to get rid of during this part of meiosis?
All the extra stuff to the cell.
Extra sets of what? Chromosomes. Those polar bodies, they're like garbage bags to get rid of extra chromosomes. Because we got to get the chromosome number down to what? 23 single-stranded chromosomes. Why not just make 4 eggs? It'll work. Turns out sperm are little bitty cells, right? Is there any way you could see a sperm with your naked eye? A single sperm? No way. Can you see a single egg cell with your naked eye?
Barely.
Turns out you can. Okay? A single female human egg cell you can see with the naked eye. No microscope needed.
Like, just so if I just like pulled out an egg all of a sudden, if I just
Why would you have one?
No,
Why
like
would you have
How are you gonna pull it out?
You have it in your pocket.
Yeah. No, but if you had a slide that was like a cross section of a woman's ovary, you could look in that cross section on the slide, just hold it up to the light, and you could start counting little egg cells in there. They're big enough to see. Why does an egg cell need to be so much bigger than a sperm?
Because it holds baby.
What do you ladies give to that baby that us guys don't?
nutrition.
The cell, extra cells.
Okay, I heard 2 things that are important, and you need to know both those things. Number 1, between fertilization and embryonic development, it takes some time for what structures that normally feed that baby to develop. The umbilical cord, the placenta, it takes time for that to grow. Which means between fertilization and the umbilical cord getting ready, what's the baby feeding on?
The egg.
What in the egg? What's the food source in an amniotic egg? What's the food source in an amniotic egg?
Yeah.
Is it the fluid? Yo.
Yolk.
Yo.
Okay? I need to put that in here. You ladies, one of the reasons your egg needs to be so much bigger than our sperm is that it's got to have a big food supply in it. A food supply that's going to last for weeks for that developing baby.
So the umbilical cord does not develop until like weeks later.
Exactly, it takes time. The other reason the egg's got to be bigger, what else? What else does your do the egg cells give the baby that the sperm don't?
Air.
That really comes in by diffusion. Organelles. Okay? All the organelles come from the mother. The sperm basically delivers its 23 chromosomes, bam, boom, and its job is done. Okay? And it's done. Its nucleus becomes part of the nucleus of the zygote. The rest of the sperm is broken down, and its job is done. The egg has to feed the baby for a long period of time, and it provides all the organelles. So it has to be bigger. So in women, let's look at how it works in a woman. Here's the setup in a woman. Here is a diploid germ cell in a woman. They're called primary oocytes. Those are your germ cells, ladies. Primary oocytes. After replication, your germ cells are going to have 46 bivalent chromosomes. Those undergo meiosis 1. Turns out that that's already happened to every germ cell you ladies have. This is over and done with, this step, for everyone. You don't have any like that left. They've all done this. And it turns out that once they did this, they freeze. They stop. They do meiosis 1 and stop. But I want you to notice something. When they split into one big cell and one little bitty cell. That's called the first polar body. Its job is to get rid of what? 23 chromosomes. It's a garbage container for 23 chromosomes. It will die. This cell is called the secondary oocyte. It's haploid, but it's got 23 chromosomes that are what shape? X, bivalent. But it turns out that this, you're frozen there right now, ladies. This is where you're all at right now. You got 23 X-shaped chromosomes in your secondary oocytes.
When does that happen?
It happens very early in life, even before birth. The only way that you ladies are going to go from here to here is if fertilization happens. If a sperm works its way into one of those eggs, it will do meiosis II. Once it does meiosis II, that forms two more polar bodies. Because this one's going to split too. And those all die. Their job, again, is to get rid of what? Extra chromosomes. So here in this egg, there should be 23 single-stranded chromosomes. That's going to get together with these 23 single-stranded chromosomes from the sperm. And that's going to make a cell with 46 single-stranded chromosomes. And we call that cell the zygote. Now, like we said a second ago, you ladies were born with a certain number of these guys, primary oocytes. What started happening to the number of those that you had before you were even born? They started decreasing. And for your whole life, that number's been decreasing, until eventually you run out.
So when are they able to start fertilizing them?
When puberty starts. When puberty starts. Yeah, because all this whole
He said they was decreasing. So
until puberty starts, they stay in the ovary, right? When puberty starts, they start being ovulated, usually one per month, and that's when the cycle starts. Yeah. Here we go.
When you like, there'll be like some type of thing where you don't get enough eggs, and then that's how like people can't.
Yeah. There's. That's not the most common thing. She's asking about like infertility. There are a lot of people who just can't have children, right?
Isn't there just like, there's also something where when girls hit puberty, instead of releasing one egg, they release like 5, and instead of having their cycle every month, they have it every 2 weeks, and that causes them to go through.
There's all kinds of weird things that can happen with the cycle.
Every 2 weeks?
Let me address Caitlin's question first. Hear me on this. The infertility thing, the two most common causes of infertility are, number one, a lot of guys have what's called a very low sperm count. They don't make many sperm. Normally, one like little teaspoon of semen has millions of sperm in it. That's important because what's it like inside a woman's reproductive tract?
They're trying to
It's
control.
quite
It's very dangerous. It's acidic, and that acid will kill the sperm. What else is coming after the sperm inside the woman's reproductive tract? Her immune system's trying to kill them. Okay, so it's like a battle going on. Okay, and the sperm are trying to make it to their goal. You know,
And they're trying to keep each other.
bad analogy here. Here's a bad analogy, but you're gonna remember this. How many of you seen the movie The 300?
yes.
When you watch the Spartans, what's their battle formation?
To line up next to each other.
It's like a triangle, like a phalanx, right? What did those guys on the outside know was their fate? They're gonna die, because their job is to protect the guys in the middle, pretty much. If the guys on the outside, they die. It sort of works that same way with sperm. When they go in like this, in a V. The guys on the outside, they're gonna die. There's a few in the middle that are gonna make it to where? Where does fertilization happen at in a woman? In the fallopian tubes. They got a long swim to get there. Okay?
They have to like jump.
No, not really. But most of the sperm, they die in the journey. Those guys that were in the middle, some of them make it. What's the head of the sperm? What's it filled with? The very tip of the sperm. Here's a word you need to know. The tip of the sperm is called the acrosomes. It's the very tip. Guess what it's loaded up with, the acrosome?
DNA.
Fuel.
Enzymes. Enzymes to do what?
Fertilizer.
It's got to do what?
Oh, it has to dissolve the
It's
egg.
got to drill into the egg, right? So those enzymes, that's what they're there for, to drill into the egg, to eat through the coatings around the egg. Because if you look at that egg, it's got a coating around it, right? It's got to drill in through there. Yeah, that's what that acrosome is for, is to drill in. Now, something else kind of cool that happens is once one sperm gets through there, what does that egg better do real fast? You better find a way to close it, because if more than one sperm gets in, you got a problem. No, not twins, 3, okay? Because we just got through saying one extra chromosome causes major issues, right? That would be a whole 23 extra chromosomes.
So you have like
And really,
is that possible?
freeze was not a good way of saying that. Yeah. What's going to happen if you got 23 extra chromosomes? It's just going to die, okay? It's not ever going to be born. So right after that first, hear me, right after that first sperm makes it through, the egg makes its hard coating around itself so that no more can get in. It very, almost instantaneously, makes this hard coat, and it blocks out any more sperm from coming in. It's kind of a cool process, the way it works. Back to what I was saying about infertility, though. It could be either a low sperm count in the guy, that's one cause. Another cause, one of the more common causes, is that a lot of times it's not the ovaries that are having the problems in the women, it's what that's got the problems? The uterus. Okay? The lining of the uterus is supposed to be nice and thick with a lot of blood vessels in it to feed the baby, right? Sometimes women develop this condition called endometriosis, where
Is that
the
hereditary?
can be where the endometrium doesn't develop the right way. The endometrium is the lining of the uterus, the inside lining, and it's just not conducive to supporting a baby. It's got scar tissue and all kinds of stuff there, and the baby, the zygote just can't implant itself the way it's supposed to into that lining of the uterus. So those are probably the two most common causes of infertility.
But wait a minute.
Go ahead.
Oh, if it doesn't like run family that's
It's possible, but I don't think they even really know why some women get it and some don't.
just feel like malnutrition going through puberty.
I don't think they really know. It's just one of those things that some people get and some don't, and we're not really sure why.
So sometimes like the zygote somewhere, plant itself somewhere that's not the uterus. Doesn't that happen? Is it?
What's
does
it
it
called when it
happen
happens
in
in the
fallopian
fallopian
tube?
tube?
All right, yep. This is the same question I got yesterday. Occasionally, remember the zygote gets formed inside the fallopian tube, which is the connection between what and what?
The ovaries and uterus.
The ovaries and the uterus. It's a very narrow tube. It's kind of windy. There are little cilia in there that are beating that push the zygote or egg toward the uterus.
So it's sort of like a assembly line.
Yeah, sort of. But sometimes that egg actually attaches up, the zygote rather, attaches to the wall of the fallopian tube. And it starts releasing chemicals that make it start to develop. And it gets stuck there. And it just gets bigger and bigger and bigger and bigger and bigger. And that fallopian tube is really small. So what does the woman start feeling? She starts feeling pain inside her. And if she doesn't do something about it pretty quickly, that fallopian tube can rupture and she'll bleed to death. That's called a tubal pregnancy. What's the only solution to that problem? Abortion. And I'm very anti-abortion. But in that case, there's two options. If you let it continue, the baby and the woman will die. There's no ifs, ands, or buts. They'll both die. If you don't take it, if you do take it, then the baby will die. There's no way of helping it, but the mother will be fine and she can have another baby later. In that case, that's about the only case where I'm for aborting a baby. Because there is no doubt in that situation that both will die. That the fallopian tube is not big enough to support a baby. It will not happen. Yeah.
How do you get pregnant and not know it? You know how like some people.
Yeah, that's
Where
what
does that show?
what
I
happens
didn't
there.
know.
I do not believe that show. Okay? I had a cousin who was very, very thin all throughout high school. And I hope she doesn't listen to this recording on Internet Explorer. She was very tiny. She was like Maddie size in high school. And she got pregnant in 11th grade. And she started gaining huge amounts of weight. Started wearing baggy clothes. And a skinny girl, there is no way you don't know you're pregnant, okay? Have you ever looked at a pregnant woman's belly? I mean, not that it's just digging around, but that it's moving, okay? I mean, it's moving. You know if you're pregnant or not. And I do not believe these shows where they look at that.
Okay, but like on the show, it's like, okay, it's like you don't gain any weight, and then you don't have any symptoms of like morning sickness
But you
or
have to
anything
wait
like that.
that.
Yeah, but, you know, here's the deal. There might be a few exceptions to this, but all of a sudden your period stops. That's kind of a good symptom that you're pregnant. I don't know that I buy that. There are some exceptions. I don't know that I buy it. But I mean, think, guys.
They said that there was women on birth control that had the 1% chance, and then so they never considered it because they were on birth control.
Right, but I mean, just think. Your period stops. You start gaining lots of weight. There's something moving in your abdomen. Okay? You crave weird foods. And I mean, cravings are not always there. But to me, having never been pregnant myself, but to me, it would seem that it would be very, 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 almost impossible not to know it if there was something growing inside you. If the period inserts. Caitlin just asked a good question. She said, how could you be pregnant and still have a period? It would be very difficult because during your menstrual cycle, what is the blood and the stuff coming out?
It's your uterus.
It's your endometrium. It's the lining of your uterus. So if that is coming out, the baby should be coming out with it, right?
I heard that it's not really a period, it's just his body.
Sometimes you can have partial bleeding from your uterus when you're still pregnant. It's not really a period, but there's some of the uterine wall that's bleeding, not the parts where the baby's at. The only way that I might at all buy this I don't know I'm pregnant thing is with a woman who was really, 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 really heavy. Okay? I might buy that. Because a really heavy woman, I mean, she may not notice the weight gain because she's gaining a lot of weight anyway. Not being correct, okay? And there's so much there that she might not feel the baby moving as much. So I might buy that. But my cousin who was pregnant, I don't buy that. She was tiny, and I just don't believe it.
Well, there was one, and it was like this woman, she got pregnant, and her uterus was like lower down than like a normal person's.
Right.
And so it didn't really form like a giant, big
Right.
belly. And so she just didn't notice herself getting bigger because it didn't look like it.
There was another one where
Yeah, but did she not notice that her period had stopped and something was moving inside her? And
Like a lot of the cases on there, they talk about like either being on birth control or like forgetting to take it or something like that. So they just thought it was.
all right.
There was one where this woman thought her uterus was falling out when she was giving birth.
Yeah.
Oh my god.
I think
She's like, I'm not having a baby, my uterus is falling out.
my general opinion on that show is that those people wanted to deny to themselves that they were pregnant. Okay, they did not want to believe they were pregnant.
And the doctors kept saying, oh, you can't have it, you can't have it.
Right.
But then it like happened because like they like one of the women on there like they've been trying for a long time, but like miscarriages, and they just said that it wasn't possible.
Right.
And she like went to like an amusement park and there were all these rides, and then popped out.
Now, every year, this is something that I really think these people I'm about to describe ought to go to jail for a long time. Every year there are girls in high school who give birth to babies at school and like in the toilet, okay? They leave them there. I mean, that's happened multiple times.
Did you hear about the girl that gave birth at college?
Right. I mean, it came out of it. You can't deny it. It's ridiculous. And a lot of times those people don't get in trouble, and I don't understand that. It seems kind of like murder, yeah. Yeah, where'd that come from? I don't understand. Anyway, before we run out of time,
Everyone noticed the price rules on.
I have a question.
you would think.
So, you know,
Guys, sit. Taylor's trying to ask me a question. Let me turn off this recording. I'm gonna have to go ahead and edit some of that out.